Okay, we're actually out of time. So so thanks again, Antoine, for this nice presentation. And we'll move to Lucy. So while you're putting up your slides, Lucy, so Lucy Gao is an assistant professor in the University of Waterloo in Canada, and she's going to talk about valid differential expression for groups defined via clustering, and especially, I mean, this is something which can be applied, as she will explain, I think, to single-cell RNA-seq data analysis. And I think it's a very important issue in the analysis of this data, because it's a problem which is often overlooked. Sorry. So Lucy, please go ahead.
Hi, thank you for the introduction, Pierre, and big thank you to all of the organizers and all of the other speakers for what has been a really great session so far. As Pierre said, today I'll be sharing a selective inference framework we developed for differential testing after clustering. So all right, let's set the stage. Let's say that we want to test for a difference in means between two groups. This is a very classical statistical problem, of course. We start teaching methods for this purpose as early as Stats 101, the first statistics class that you take. Of course, there's an important caveat to these methods, though. And that caveat is, well, we have to define these groups before we look at our data. And this can be fine when the groups are different experimental conditions. But what if we actually want to define these groups by instead doing something data-driven, like applying a clustering method to our data? If this is really what we want to do, then what we're really asking to do is, speaking to the title of the session, is we want to double dip. What we really want to be doing here is we want to be able to both use our data in order to generate some kind of a hypothesis, and then use it once again to really ensure that whatever results we get by testing this hypothesis is truly interesting. And perhaps the archetypical modern example of this task, though it occurs in a lot of places, comes up in single-cell RNA sequencing analyses, which I'm sure a lot of you in the audience are more familiar with than I am even. But I'll just briefly go over. A common analytics task in these settings is you would like to be able to compare the gene expression profiles across different cell identities. Unfortunately, for various reasons, pre-sorting cells into different cell identities is not always possible, or in fact, even usually not possible. And because of this, virtually every computational pipeline out there is going to first start by assigning, like putative cell type labels by first clustering these cells, and then using that same data in order to test for differential expression across the clusters. So what is our task here? Well, okay, so let's say you give me some kind of a data set. I visualize it as 2 features, but of course you can think of this as the first 2 principal components or something like that. And given this data set, what I want to do is I want to go ahead and I want to cluster it using some clustering algorithm. And here I've chosen hierarchical clustering, and I've just decided to cut the dendrogram at a particular level in order to get 3 clusters. And now I want to test for a difference in means between these 3 clusters. And at this point in the session, I'm sure you have some kind of a suspicion that, like the naive approach where you just apply some kind of a classical test for a difference in means is not really going to end well. But just how bad is this naive approach? I'm going to go through some simulated data in order to find out. So what I have shown on the slide is what I've done is I've done something fairly simple. All I've done is I've just sampled 100 observations from a, like a standard bivariate normal distribution. So there is really no signal from this data. Every one of these observations has been generated from a distribution with the same mean. Even so, I'm going to go ahead and I'm going to ask my clustering algorithm for 3 clusters. And of course, I ask, it'll oblige, and it rather uncritically will humor me by supplying me with these 3 clusters. So now that I have these clusters that my algorithm has given me, what I want to do is I want to calculate a p-value for a difference in means between each of the 3 pairs of clusters. All right, all well and good, but before I even calculate the p-values, I have a suspicion that there's something funny going on here. Because if you look at the distances between the cluster centers, they're pretty far apart, right? These cluster centers are not really close to 0 at all. So given this visualization, perhaps it's not a big surprise that all 3 pairs of clusters turn up super tiny p-values for a difference in means. And this is a pretty bad result to be having because remember, this data set here, I literally just simulated standard bivariate normal data, right? All of the observations literally have the same mean. Yet these really tiny p-values are suggesting that these observations in these different clusters actually are very different. And all right, that was just one single simulated data set. So to prove to you that I haven't just gone ahead and somehow cherry-picked a very terrible example, I have here a QQ plot of 2,000 simulated data sets using the same kind of settings. And on the x-axis here, I have quantiles of the uniform 0-1 distribution, which we know is the theoretical distribution of a valid p-value. And on the y-axis, I have these empirical quantiles of the naive p-values. And if all is well in the world for a statistical inference, we would expect these p-values to be all on the red 45-degree line, but instead I have this pretty crazy looking swoopy line at the bottom. So what this is telling us is that, well, the naive p-values, they're not uniform. They're actually pretty far from uniform. What's even worse if we think about what direction they are in comparison to the p-value, to the uniform distribution. And in fact, largely speaking, my p-values are ridiculously tiny compared to the uniform distribution. So as it turns out, what this means is that if you use, like the standard significance level of 0.05, even though I simulated truly null data, I'm rejecting the null hypothesis of a difference in means between clusters literally like 97% of the time, which is, 97%, this is an astronomical number. I knew that naively performing a test for a difference in means was bad. I have to say that I was actually surprised that it was this bad. Look at this, we're virtually guaranteed a type 1 error. So this definitely suggests to us that a naive test for a difference in means isn't really going to solve our problem here. But all right, maybe we expected that. That was, of course, the simplest possible solution. What if we do something a little bit smarter, like something involving sample splitting? You might think, well, surely if we use some kind of a test set, this problem is just going to disappear. Well, all right, let's go ahead and try it. So what I have on the left here is I have that same noise data set, no true clusters. But this time I have made half of them displayed as squared as my training set and half of them displayed as triangle and made them my test set. And I'm going to go ahead and then apply a clustering algorithm to the training set only. Then I'm going to go ahead and I'm going to train a classifier on the training set, and I'll use that to assign or port labels to the test set. And in particular, I use 3 nearest neighbors classification, but of course the details of the classification algorithm aren't the important part here. What's really important is that we're actively running the clustering algorithm on the training set only. We're not directly running it on the test set. But does this really solve our problem? Look, if you look at the empirical means in the test set, the mean of the orange observations is, again, nowhere close to the mean of the green. So once again, if you perform a naive test for a difference in means, you get this very tiny p-value. So what happened here? This is at least a little surprising. But if you think about it a little bit harder, it does actually make a sort of sense. So why do we expect sample splitting to work? Sample splitting usually works in a lot of selective inference type settings because we can avoid double dipping by using sample splitting. But unfortunately, in this case, we weren't actually able to avoid double dipping at all here. And the reason is we actually had to look at our test data, though perhaps it's a little bit less obvious, in order to apply these cluster labels before testing for a difference in means. That's still double dipping. Step 3 here is generating a hypothesis using the test set, and then step 4 is testing it. So clustering is this unusual double dipping problem that can even be solved by sample splitting. Even if you use some kind of a sample splitting approach, you're still going to inflate your type 1 error rate. And in the rest of this talk, I'll propose a general framework for testing for a difference in means after clustering without specializing to any particular clustering method. Then we'll talk about the computational aspects of this test in the special case of hierarchical clustering. So let's start by putting our questions in more formal statistical terms. We're modeling our data set as independent draws from a multivariate normal distribution, where the ith observation has this mean mu I, and every observation has this known isotropic covariance matrix. We're going to imagine clustering that data set in order to get k estimated clusters. And given that we've done that, we can associate a sort of mean parameter to each of those clusters. So these mu bars, the k's, are just the population mean that's associated with the observations that were assigned to the kth cluster in my clustering procedure. I think this is a fairly natural sort of way to define a mean for the kth cluster, since this is just a group, I'm just taking the group empirical mean. But it is sort of like a funny sort of parameter in the sense that the mu i's, these are population means, they don't depend on our data at all. But because we're averaging these population means, these mu i's over observations assigned to an estimated cluster, where the clusters are estimated from our data, we end up with this mean parameter that depends on our data. And once we have these means defined, the null hypothesis looks pretty familiar, which is just that there's no difference between the mean parameters associated with 2 estimated clusters. So if you wanted to test this null hypothesis using a classical test, what we could do is we consider using the wald test for a difference in means. And this would be under the normality assumption, this would essentially end up with a p-value that has a theoretical chi-squared distribution. And this is how you would calculate the p-value. You can just use the theoretical quantiles. But unfortunately, as we saw in our simulations, this p-value is not going to be uniformly distributed under the null. So how am I going to fix this? Well, I'm going to claim that I can fix this problem by conditioning on the way that we selected the null hypothesis. Instead of defining the p-value to be, the probability of seeing such a large difference in means under the null, I'm going to define it to be the probability of seeing such a large difference in means under the null, but this time conditional on having estimated these clusters C at k and C at k prime. if you unpack this p-value a little bit to try and interpret it, you do get the statement that should look very familiar to how we would interpret the standard wall p-value. In fact, there's literally exactly one difference, which is just whether you have this wording in blue or not. And this says out of all data sets that would result in the clusters C at K and C at K prime. This also gives us sort of a sense of why this correction will work while the naive test doesn't. We're basically holding our results to a higher level of evidence. The wall test is comparing what we're seeing in our data to all data sets, which seems silly. You know, there's a lot of data sets out there that, in particular null data sets, that are not going to result in the clusters that we saw that have pretty small difference in means because they're under the null. It's a pretty easy bar for a data set which did result in these clusters to clear. A much harder bar is to compare against all data sets that would have resulted in these clusters, which, you know, if our clustering algorithm works at all, would tend to have a somewhat larger observed difference of means. And this corrected p-value controls the selective type 1 error rate, which is basically the error rate of our test, given that we decided to test this null hypothesis that our data shows, and also that that null is true. And this is essentially because we chose to test this null hypothesis because we estimated these clusters in our data, and that's exactly what we're conditioning on in the p-value definition. All right, sounds good. If we could compute this p-value, then we would be good to go. But unfortunately, as it turns out, it's pretty hard to compute. And basically what I mean by that is it's difficult to write down an explicit formula for the null distribution. For one, it depends on nuisance parameters that aren't known under the null. So this is good in theory. Unfortunately, we can't literally calculate it. So instead, our idea is just to condition on a little bit more information. And the motivation for this is if we're clever about what kind of extra stuff we pick, we can make our computation a lot easier. And crucially, even though we're conditioning on more, we're still going to be able to control selective type 1 error. So what is it that I'm actually conditioning on? What is this more stuff? I've written it out explicitly here, but you know, if you look at it, perhaps this looks like a random jumble of linear algebra. It might seem a little bizarre, but please bear with me for a couple more slides. We are actually conditioning on something fairly interpretable, and we'll see more on that in a couple slides. And the key to understanding this p-value that we've defined is this result. It's basically saying that this p-value is actually equal to the probability that this univariate random variable phi, which I defined to be the norm of the difference in means, is larger than the difference in means that we observed, given that this x prime of phi thing, which is an n by q data set that I haven't told you about just yet, will yield the same two clusters that we saw in our original data. And what is this n by q data set, x prime of phi? Well, it's essentially constructed by perturbing our observed data. And this is the mathematical definition, but I wouldn't look too closely at it because it's much easier to explain with a picture. So to get x prime of phi for a given phi, what you're really doing is you're just taking your original data, little x, you're drawing this line through the empirical means of the two estimated clusters. And then if phi is larger than the distance between these empirical sample means, all you're doing is you're just taking these observations in the estimated clusters C at K and C at K prime, and in a single piece, you're just pulling them apart along the axis of that line. And if phi is smaller than that distance, then you're taking these observations and this time you're pushing them together, again on the axis of that same line and in one single piece. And if phi is the same as the distance between those sample means, well, you're not doing anything. You're literally just getting back the original data set. So to summarize, this x prime of phi is just a version of the original data set x where what you're doing is you're just either exaggerating or de-emphasizing the difference between the estimated clusters by pulling them or pushing them together. And even though we had that weirdly complicated conditioning set, it really just reduces to the simple statement in terms of phi. And it's really, again, it's just asking the same question as the standard Wald p-value, except instead of out of all n by q data sets in the universe, we're just considering the set of perturbed data sets x prime of phi that if you cluster them, you would get back the same original estimated clusters c at k and c at k prime. And what's more, it's now pretty straightforward to write down an analytic expression for the p-value. And you can basically calculate it using the CDF of a random variable that's proportional to a chi-squared, which you've cut off outside of the set S, where the set S is defined to be the set of phis that would create a perturbed data set x prime of phi that would still result in these same clusters c at k and c at k prime. And as long as we can calculate the set S, it's pretty easy to calculate this p-value using the CDF of a chi-squared random variable. So there's really only one question left, which is how are we going to compute this set S? Well, up until now, everything really applied regardless of what clustering method you apply to your data. But calculating S for any type of clustering is a bit of a difficult problem. So starting from now, we'll focus on a smaller problem, which is a specific type of clustering, hierarchical clustering with squared Euclidean distance. I don't want to go through too many of the computational details, so I'm just going to try and sort of give you sort of the flavor. The critical idea that lets us compute this conditioning set is basically, as it turns out, if you cluster a perturbed data set x prime of phi, and you get the same cluster c at k and c at k prime, not only do you preserve those two clusters, then you actually preserve the entire hierarchical clustering dendrogram up to the merge that created these clusters. So visually, the idea is that, you know, if your original data looked like this with this dendrogram, then something like this on this x prime phi on the right, where you end up with the same cluster assignments, but with a different merge order, this isn't possible. And this is really the key result that gives us a strategy for how to explicitly compute this set S for hierarchical clustering. And the basic idea is we look for x prime of phis where the hierarchical clustering algorithm is going to do the exact same thing up to the n minus kth step. And that allows us to rewrite this as an intersection over n squared set, basically all of the cluster pairs that lost and were not merged in each of the n minus kth steps. And it turns out that for many linkages, each of these sets turns out to be the solution set of a quadratic inequality, which can be solved in constant time. So what are the implications of this? This means that for these linkages shown, we can actually calculate this set S in quadratic time. And the argument is a little bit different, but you can also show that you can calculate the set S for single linkage in quadratic time as well.
Lucy, just interrupting to say that we have a bit more than 5 minutes left, including questions. So, I mean, just to let you know, because I would like to save like at least 3 minutes for questions. So.
Okay. Okay. I'm sorry that I am running over time. Perhaps I will just skip to, I have the simulation results, but simulation results aren't usually very interesting. They basically say that the p-values are in fact uniform. And just to try and wrap up to the part that you're probably going to be most interested in, I have an application to single cell RNA sequencing data. And basically what I did was I'm working with this Zhang et al. Data set, and I subsampled 600 cells that were labeled as T cells to serve as a negative control. And I subsampled each of 3 different cell types in order to serve as a positive control that seems to have clusters. And we're going to apply our test and see what happens in each of these cases, 1 cell type versus 3 cell types. And first, when I cluster a single cell type and I apply word linkage hierarchical clustering, you can see there's perhaps like a little bit of separation on the PCs, but this isn't perhaps super clear separation. If you apply the naive wald test, you're just going to get these super tiny p-values, while our selection adjusted p-values are much larger. And if you apply word linkage hierarchical clustering to the mix of cells, you get these estimated clusters that do line up with the true cell types. And while the wald p-values are pretty small, our p-values are pretty similar, which suggests that we are maintaining some reasonable level of power. And again, apologies for running a bit over time. If you want to learn more, of course, our preprint is out, and we have an R package as well as a website with a nice little tutorial. And a big thank you to my collaborators on this work, who have been a blast to work with. And I would love to take some questions now.
Thanks a lot. Sorry for rushing you, but it's good that you had time to cover the single cell RNA-seq application. Okay, so we have a few minutes for questions. There is a first questions by Thomas, but so I'll read the question because he answered himself to his questions. I will read everything. The fundamental hypothesis of clustering is that you have several subsets of the population with different distributions, while the null hypothesis states that they are not. Thus, it looks like piping a difference of mean test after clustering is an answer to an ill-posed question. And then he commented and said, okay, in fact, the conditioning you introduced to compute the null probability looks like you're correcting this ill-posed question.
Yeah, that's a really great point. I mean, it really is just that, okay, like once you've clustered your data, is the distribution of what you observe after you've like done the separation, is it going to be different? I don't need to do a hypothesis test to do that. Of course it's different. Like you've done some filtering. So really, like that's why like a naive test of a difference of means is going to, that's one way to think about it. Like the naive test for a difference of means is really asking the wrong question. While we're interested in, okay, like, is there a difference of means in the population parameters? Like are we in this case, we're asking a more reasonable question, which is, once we've done the selection, the distribution is different, but was there a distributional difference in the observations before this clustering selection procedure?
Thanks. So I don't see more questions in the Q&A. So maybe so feel free to ask to raise your hand, or maybe among the speakers. I mean, I have questions. I can ask one. So for example, my first question is, so in your to make your method tractable, you introduce a second layer of conditioning. I mean, you're conditioning more, as you said. So what is there a price to pay for this in terms maybe of power?
Yeah, you definitely pay a price in terms of power. And one way to think about it is you're sort of like artificially excluding relevant null data sets, right? Really what we want to do is we would just want to consider all null data sets where we would have selected the same null hypothesis. We're sort of artificially like limiting the universe of null data sets that we're considering. So it's reasonable to think that we might be losing some power. That being said, though, I do think it is worth it but it's introducing this extra layer of conditioning, conditioning on this orthogonal subspace and conditioning on the direction. That's really what enables us to analytically compute the selective inference p-value, which if you don't make that assumption, you can still probably come up with some kind of a Monte Carlo approach based on sampling. And we do propose like a version of that in the paper where we approximate the p-value using sampling, but that's a little bit less elegant in the sense that you're always going to have an approximation, and it's also computationally expensive. So it's really a trade-off thing here.
Thanks. So we have a question from coming from Nancy. Could your method be used to determine the optimal number of clusters?
Yeah, we've thought about this a little bit. One of my collaborators, Jacob, actually is particularly excited about this, especially in the context of hierarchical clustering, where, you know, the standard practice is just to like cut the dendrogram like entirely horizontally at a certain point. But it might be nicer to be able to say like, well, this particular cluster at this level looks nice, and this particular cluster at a different level looks nice. And one thing that we've thought about is starting at the top of a hierarchical clustering dendrogram and like testing, well, okay, at the first split, is there a difference in means across that split? If so, let's keep going and then all the way down. Of course, you then have to apply some kind of another layer of correction in order to account for this hierarchical selection of null hypotheses. But in principle, if you do that, it could be an interesting way to choose the number, to get a set of clusters from hierarchical clustering that are certified in some way by significance.
Yuval, yes. Last question.
So thank you for the great talk. In some sense, what happens if we have a cluster where one example is from one type and everything else is kind of the same? Somehow I think, right, the null model assumes that everything in the two clusters is from the same distribution. But if we have the split to be slightly different, then we're still not in the null case, right? So even though I had one example that was, that was different, somehow was tagged into one of the clusters, already I'm deviating from the null model. So is there any sense of, I guess the continuing question is there any sense of effect size that we can give here that says like how many within the cluster are from the same mean or something like that?
Right, of course that's true. You know, like as you said during your talk, we never really expect the null model to be exactly true. In your particular example, like where one observation has a different mean, but like the remaining, like 19 observations in an estimated cluster have the same mean, you would expect the you would expect a relatively like you wouldn't be literally exactly under the null, but you should be pretty close to the null. So some measure of effect size or some kind of a confidence interval would be really nice here. Theoretically, it's a little bit challenging to construct confidence intervals in this setting, just because as it turns out, even though like the theoretical conditional null distribution is chi-squared, when you consider the null of no difference in means, if you then consider, if you want confidence intervals, you now have to consider, well, the null hypothesis of a difference in means being equal to some non-zero value. And as it turns out, that you don't end up getting a truncated chi-square distribution under the null anymore. That being said, Rena Barber's group has a workout on selective inference from the group class. So I think from like 2016, and I think they showed there that even though you can't construct a confidence, it's difficult to construct a confidence interval. What you can do is you can construct like a confidence lower bound on the size of the effect. And I think that's something that could be applied here, though I haven't actually executed on it.
Thanks.
Okay, thanks a lot. So I think we're a bit over time, so we need to close the session now. So I'd like to thank all the speakers, of course, for their very nice talks. I hope this has been hopefully inspiring for applications to bioinformatics. This hasn't been mentioned systematically, but I think all the methods that have been proposed are implemented and available. I mean, I guess the one of Antoine is not finalized yet, but I'm knowing the co-authors, I know that it will be available and usable. Yeah, and also I would like to thank the audience, because I mean, you've been a very large audience. I counted 127 not people but connections, because some of them were multiple connections. So I think you can say it was a very successful session. So thanks a lot to everyone, and well, see you tomorrow for the rest of the conference.
Thank you very much. Thanks, Pierre, and everybody for setting this up.
Thank you. Thanks for staying late, Yuval, because it's later in your
just
place.
1 hour, yes.
Okay, and I don't know what was the rule is, but if people want to stay around and interact with the speakers, we can, if the speakers are still here. So typically, if Lucy has time, and if someone wants to ask a question, you can be promoted, I guess. I don't know if it's a bit late in France, so I don't know if this will work out. So maybe I can ask you another question. Another question I had, Lucy, which was, so I was actually very surprised by the fact that the sample splitting didn't work at all. I mean, it's just like it didn't work really. It worked really bad. So I was wondering whether it could also. I mean, you gave some possible explanation. I was wondering whether it was also possibly due to the fact that you are looking at. At a small example in two dimensions. So I was thinking that maybe the problem with sample splitting would be less important for example, for single cell RNA-seq data. So have you tested sample splitting? I think there have been papers on using sample splitting for differential analysis after clustering for single cell data.
Yeah, that's a good question. I haven't explicitly tried it on higher dimensional data, but the papers that you mentioned that focus specifically on a selective inference approach for to deal with the differential expression problem for single cell data. I'm thinking of particularly this paper from Zhang and others from, I think it was 2017, valid post-clustering differential analysis for single cell RNA sequence data. They certainly, in their simulations, they definitely used less higher dimensions, and they ran into exactly the same problem. In general, I actually think that the more features you use, the almost like the problem becomes more severe because you're selecting on because you're like selecting more aggressively. Like there's like, it's not just you know, like when you cluster, you're not just going to see like a larger than zero difference on one axis, like a single features axis. You're now seeing it on like 100, you know, like say 100 or even 1,000 axes. So I think it might even make the problem worse, but I haven't explicitly tried it.
Yeah, I mean, I would be curious. I agree with your intuition. That's, I mean, the higher dimensions, the high number of dimensions, basically the more likely you can find false positive clusters, basically. So then, yeah, okay. So that's, but that's very surprising. I mean, this numerical example was very, very striking to me. So, but I don't have a sense of what it I mean, I haven't tested your method yet, so I don't know exactly. I don't have a computational, if you want, a sense of what happens. Okay, well, thank you very much for the talk again, and yeah, so we hope to be able to work on related questions soon. But it's, I mean, we've looked at your paper, and it's not an easy topic. So I think it's a very nice work that you presented.
Thank you.